Hi everyone, welcome to the monthly E3SM BGC webinar. Our speaker for this month is Dr. Lisa Welp, who's a professor at Purdue University, and she's going to talk to us about using stable carbon isotopes to constrain terrestrial biosphere water use efficiency. Thank you, Lisa.
All right, so today we're going to talk about using stable carbon isotopes to constrain the terrestrial biosphere water use efficiency. So just as a kind of a little introduction here on the slide, I'm going to be talking about different types of CO2 in the atmosphere. Most of the CO2 in the atmosphere, about 400 ppm, is what we're going to call this light carbon, with carbon mass 12. And there's about 1%, or only about 4 ppm, that has this heavy carbon isotope in it. And so this naturally occurring, just a little bit present carbon isotope in the atmosphere can give us really interesting information about the terrestrial biosphere. Okay, so I just wanted to give you a little bit of an introduction as to my research. I have a very interdisciplinary research focus. I'm focusing on vegetation, water, and climate interactions. That's fine. So my research focus, there's kind of three themes: hydroclimatology, the sensitivity of the terrestrial carbon cycle to a change in climate, and then this third theme of plant water use efficiency. In a couple months, I get to craft a presentation where I talk about all these things, but today we're just going to talk about plant water use efficiency. Okay, there we go. So since I know that this audience is mostly terrestrial carbon cycle modelers, I thought I would frame this from a modeling perspective. So this is a review paper by Purdue in 2016, and it summarizes the key knowledge and data gaps for modeling the influence of CO2 on the terrestrial carbon sink. And so today we're going to focus on a couple of these, which are highlighted in red, as areas that we need to understand better. And so we're going to talk about stomatical conductance, which links the carbon cycle to the water cycle. And also I'm going to touch on the allocation of photosynthesis. Kate, I don't know if you can let me know, can you see my cursor at all or not?
Yes, we can.
Okay, great. Thank you. So leaf stomata are these little holes in the plant leaves that regulate CO2 and water and heat exchange with the atmosphere. These are very, very responsive to light intensity or changes in light intensity, also drought stress, both through atmospheric demand and soil moisture conditions, as well as atmospheric CO2 concentrations, and also to some extent nutrient status, and different species have different stomatal control as well. These are very sensitive to climate change, and they are predicted to be a major source of uncertainty in predicting future climate. So it's important that we understand their behavior a little bit better. Some recent publications have shown that photosynthesis may have increased by about 30% over the 20th century. So this is work by Campbell, published in 2017. And then in addition to that, there's this study by Chang, also published in 2017, that suggests that this increase in photosynthesis came at the expense of little extra water use. And so both of these things, an increase in photosynthesis rates and an increase in water use efficiency, are both attributed to the increased CO2 concentrations in the atmosphere. Of course, plant water use efficiency also has important consequences for the global hydrologic cycle, as well as the carbon cycle. So this is a figure from the IPCC AR5 report. And it's just to highlight, how increase in CO2 concentration can change important components of the hydrologic cycle as well. So there's important motivating questions remaining. So how does the terrestrial ecosystem water use efficiency change with CO2 concentrations, and then also with drought? How will evapotranspiration and precipitation change with climate change? And how do carbon-water feedbacks influence net carbon sinks? So today I'm going to talk about stable isotope techniques, and these are particularly well suited to track stomatal level response from the leaf scale and the plant stomatal scale all the way up to the global scale. So I love this slide from Dennis Baldacci, which shows that these spatial scales involve 14 orders of magnitude range, right? So we're going to talk about the full range of this spatial scale today. So a little bit of primer on stable isotopes. Isotopes are the same element with a different mass because of extra neutrons. So since today we're talking about carbon, here we have carbon mass 12, which is composed of 6 protons and 6 neutrons. And then carbon mass 13, which has an extra neutron for a total mass of 13 AMU instead of 12 AMU. So the C13 carbon atom is heavier, and so we'll use that whenever I talk about stable isotopes. So the heavier isotope versus the lighter isotope, referring to carbon 12. And again, the C13 only constitutes about 1% of CO2 in the atmosphere, but as you'll find out, it's particularly useful. This is kind of a generic isotope slide that I have in most of my presentations. Again, we're only going to talk about carbon today, but I do also do a lot of work in water using stable isotopes as well. But I just wanted to give you a little bit of an indication that there are different measurement techniques for how to measure these. So the ones that I'm going to talk about today, atmospheric CO2 and then plant carbon, which is combusted to CO2. Both of those techniques introduce carbon dioxide gas into an isotope ratio mass spectrometer, and the different masses are physically separated. And so then we can count the relative abundance of each. So we're interested in the heavy to the light ratio. And because these are small numbers, we reference them to an agreed upon international standard. And because the numbers are so small, we multiply by 1,000. So you're going to see a lot of the data expressed in what is called delta notation in units, if you will, of per mil. And again, we're going to talk about these two different CO2 types with carbon mass 12 and carbon mass 13 today. So the carbon isotopes of atmospheric CO2 in plant tissue can be used as an estimate of water use efficiency because they're sensitive to the stomatal opening size and the ratio of the CO2 concentration inside the leaf to the atmosphere. So the way this works is the lighter CO2 diffuses faster into the leaf, and then it also reacts faster with the enzymes during the process of photosynthesis. So if we think about this, so here this box is the atmosphere, and this box on the right is the leaf. The leaf tissue has relatively less of the heavy carbon, and the atmosphere then has a slight accumulation of this trace amount of heavy carbon, or C13. And the difference, so this offset between the ratio of C13 to C12 in the leaf and C13 to C12 in the atmosphere, is dependent on this leaf gas exchange. So there's a couple more slides to explain it in a little more detail. But in summary, photosynthesis favors this light carbon over the heavy carbon, and the magnitude of this difference, so the amount of discrimination, if you will, depends on the Ci over Ca concentration. So this is a simplified equation that we can use to understand the C13 discrimination by photosynthesis, which you can think of it as approximately the difference between the delta value of the plant and the delta value of the atmosphere. Depends on a term related to diffusion of CO2 through the air and enzyme fractionation against C13, and then this ratio of CO2 concentration inside the leaf to the atmosphere, or Ci over Ca. This is a simplified version of the equation just to get you kind of used to it. Later on, I'm going to add a few more terms to it just to prepare you. So these C13 measurements, either a plant tissue or atmospheric CO2, can be used to estimate intrinsic water use efficiency. So here's that same equation we have. And I'm going to go ahead and introduce intrinsic water use efficiency. So this is the ratio of the photosynthetic flux divided by the stomatal conductance. And so if you use this term for photosynthesis here, you can simplify this into a term that's related, that is the atmospheric CO2 concentration, and a term that is the Ci over Ca ratio of the plant. So you can think about this as the physiological response of water use efficiency, or the part that's controlled by the stomata and not controlled by the vapor pressure deficit or the atmospheric demand. So to look over here on this diagram on the right, the atmospheric delta C13 value is about -8 per mil. And then we have plants. There's two different photosynthetic pathways shown here, the C4 photosynthetic pathway and the C3 photosynthetic pathway. And so both of these plant tissues are lighter. They have less of the heavy carbon. So here the more negative values indicate less of the heavy carbon in the plant tissue. Right? So this difference between the atmospheric value and the plant tissues, this is approximately the isotopic discrimination. So C4 plants have a small isotopic discrimination. That's because they have a low CI over CA ratio and a high intrinsic water use efficiency. C3 plants, by contrast, have a larger photosynthetic discrimination from the atmosphere. This is because they have a higher CI over CA ratio and relatively lower intrinsic water use efficiency. But you can see within these plant distributions, right, there's quite a range of CI over CA values and hence a range of water use efficiencies. So today I'm going to kind of break this up into two different stories. One is using atmospheric investigations, so looking at trends in stomatal conductance due to increased atmospheric CO2 concentrations. And for that, I'm going to present some data from the atmospheric CO2 record from the Scripps Institution of Oceanography record. And then the last bit of the presentation, I'm going to talk about investigating this in plant tissues. And so looking at the role of carbon allocation in interpreting plant tissue C13. So we'll touch on both of those today. So the first one addresses the question of how the terrestrial biosphere has responded to increasing atmospheric CO2 concentrations and climate change over the past few decades. And so this work was published, Ralph Keeling was the lead, in 2017 in PNAS. So stomatal aperture is sensitive to atmospheric CO2 concentrations. We know this, from paleoclimate records, but also, or paleo-ecological records, but we also observe it now with the increasing CO2 concentrations in the atmosphere. So under high CO2 conditions, the plant stomates can close a little bit, or they can, reduce in size. They don't totally close. And this can be an advantage in a water-limited environment. So if there's higher CO2 concentration in the atmosphere, they can still get enough CO2 through this opening, but at the same time reduce water loss. And so that improves intrinsic water use efficiency. There is some concern, though, that, however, water loss from a leaf is sort of their natural air conditioning, and so this might increase temperature stress for the plants. So when plants remove CO2 from the atmosphere, the residual delta C13 of the atmosphere increases. So let's just zoom in a little bit on an average seasonal cycle. So we have from January to December, we have CO2 concentrations in the atmosphere. Spring comes along, the terrestrial biosphere removes CO2 from the atmosphere. And at the same time, the delta C13 value of the atmosphere increases because plants are removing that lighter carbon, right? And so the heavier carbon in the atmosphere becomes enriched. So this seasonal cycle is due to the terrestrial biosphere. So just keep in mind a little bit later that this atmospheric signal that I'm going to talk about is kind of the inverse of the plant tissue signal that we'll discuss a little bit later. So here, you'll see this again. This is CO2 concentration in the atmosphere measured at Point Barrow, Alaska. So you guys are familiar with CO2 concentrations in the atmosphere increasing. There's a seasonal cycle that's due to the terrestrial biosphere uptake and release. And then we have kind of an equivalent C13 seasonal cycle also driven by terrestrial biosphere activity. These long-term trends, however, are dominated by fossil fuel emissions. We'll talk about that a little bit later. They also pull down the delta C13 in the atmosphere. So this first story I'm going to tell you comes from long-term records of atmospheric CO2 and delta C13 change in the CO2 flask network. So since 1950s, the Keeling curve record has been measuring CO2 concentration increase in the atmosphere. But since the 1978, they added on this isotopic measurement of the atmospheric CO2 as well. So this flask network is called, consists of a number of stations in a transect through the Pacific. Ralph here is holding a glass flask that is used to sample the air. So these flasks are evacuated in the lab. So there's a vacuum inside of them. All the air is pumped out. And then whenever they are deployed at the field, I'm going to mostly talk about data here from Point Barrow, Alaska on the North Slope. When it's time to collect a sample, someone goes out and opens the valve and air rushes inside the flask. That is then closed and brought back to the lab, and CO2 concentrations are measured, and the CO2 itself is extracted and then analyzed for the carbon isotopic ratios. So that goes back to 1978, and then we have ice core records before that, and so you're probably familiar with these. You can drill the ice cores, and the little air bubbles inside the ice can be used as a record of not only atmospheric CO2 concentrations, but also C13 in that CO2 going back over time. And so this is an updated figure, so published in 2013. The more recent data here is from the flask network, and then the older data is from the ice core records. So as CO2 concentration has built up in the atmosphere, the C13 concentration has decreased as well. And so this is one of the sort of smoking guns that we know that current climate change is driven by fossil fuel emissions to the atmosphere. Because fossil fuel carbon is an old plant, it has a really light C13 signature, right? So that's been pulling it down over time. Okay, so I'm going to talk about two ways of interpreting the C13 of CO2 record. One from the long-term record that was published in PNAS, and one from the annual seasonal cycle, which is in preparation. So first we'll talk about the long-term record. So this image here, get my cursor to work. So here we have the delta C13 observations in the ice core, where the data is kind of sparse, and then it transitions into the flask record since 1978. And so then the data is much more dense here. So if we were to assume that the C13 discrimination of the terrestrial biosphere had not changed over this time, then you would expect the black dashed curve here. I really hope you can see my cursor. Right, which decreases much more rapidly than the observations. So in order to explain the slower decrease in delta C13 in the atmosphere, we have to invoke a change in the terrestrial biosphere discrimination over time. And so you can look at this top panel up here. So these three blue curves are all different ways of estimating a change in the terrestrial biosphere discrimination over time. The solid one is based on CO2 concentrations. The dotted one is based on temperature increases, and the dashed one is just kind of it stays constant and then increases kind of arbitrarily. But all three of these, some increase in photosynthetic discrimination, match the observations much better. So what we take away from this is that there has to have been an increase in photosynthetic C13 discrimination of the terrestrial biosphere over this time, probably due to increases in CO2 concentration in the atmosphere. This is a full equation. It's a simple one that I showed earlier with photosynthetic discrimination being a function of the Ci over Ca concentration, but it has some other terms in here as well. And I'm going to come back to these in a little bit. But so this indicates an increase in photosynthetic discrimination over time. Then we can look at the seasonal variation and extract information from that as well. So again, we have CO2 concentrations with a seasonal cycle increasing at Point Barrow, Alaska. And then this is the delta C13 of that same CO2 sample, also at Point Barrow, so which has been decreasing but also has this seasonal cycle. So again, the seasonality is dominated by the biospheric activity. The long-term trends are dominated by fossil fuel emissions. But we can use this covariance to extract additional information. First, I guess I'm just going to convince you a little bit more that the seasonality in CO2 concentration in C13 is dominated by the terrestrial biospheric activity. So here we have 2 sites, Point Barrow, Alaska, and Alert, Canada. We have the seasonal cycle of CO2 concentration and C13. The observations are in black. You can see they have a very large seasonality. And then what's also on here are predictions of the influence on the seasonal cycle from fossil fuel emissions, from ocean gas exchange, and from something called isotopic disequilibrium, which I probably won't have too much time to go into today. But you can see that they contribute very little to the seasonal cycle. They have almost no effect on the seasonality, but they will become important when we look at quantifying small trends in photosynthetic discrimination over time. So this is how it works. So for each year, we take the observations and we can plot the difference between the CO2 observations, so the concentrations, and the background value. So that's what's plotted on the x-axis here. And then we can also take the product of the delta C13 and the CO2 concentration, take that for the observations, subtract it from the background trend. When you plot those, then the slope is the isotopic composition of the source. So here this can be thought of kind of the isotopic composition or the isotopic signature of photosynthesis. Because we know how the atmospheric C13 value changes, we can take the difference between this inferred delta source for each year and what the background atmospheric delta C13 value is, and we can calculate what I'm going to call an apparent C13 discrimination. So this is similar to what we were talking about before. This approximates the land C13 discrimination or the terrestrial biosphere discrimination, but with a few caveats that are going to come up in a minute. So we can calculate the slope fit for each year to see how C13 discrimination changes over time in a much more detailed interannual way than what we saw with the long-term trend. So here we have the top panel is information extracted from the Point Barrow record, and on the bottom panel is information extracted from the Alert Canada record. And here I've just shown the pink is the Scripps record, and the blue is the same sort of data that's measured by NOAA, both CO2 concentration and C13 of the atmosphere. And so I've put that comparison on here as well. So you can see that the mean value in the discrimination is about 19 per mil, which is consistent with the dominant C3 photosynthetic pathway. But there's a lot of year-to-year variability, and we're still working to figure this out. It's likely caused by regional droughts or differences in air mass influences, and particularly for these points here in the NOAA record, I think it's due to differences in data screening approaches. Okay, so if we just look at the Scripps record now, if we can fit a linear regression through this, the trend at Point Barrow is about 0.17 per mil per decade. And I'm going to explain this in a little more detail, and then we're going to compare it to the long-term record observations as well. So I mentioned this is the information that we extract just from the atmospheric variability, and this I'm going to call the apparent isotopic discrimination. But the atmospheric delta C13 value is not only controlled or influenced by the terrestrial biosphere exchange, so photosynthesis and respiration. It's also influenced by fossil fuel emissions and ocean gas exchange. And so we can make corrections for each of these in order to solve for the isotopic discrimination of C3 plants, in this case for the high latitudes. So we can take our parent observation trend, we can correct for the ocean influence, the fossil fuel influence, and then in this case, because we're looking at northern hemispheric records, there's been a strong increase in maize production, agricultural maize production. And because it has such a different C13 value, we also make a correction for that as well. So after we apply all of those, we get an actual change in the C13 discrimination for high latitudes from 1985 to 2017 of about 0.25 per mil per decade. Okay, so we can compare what we've derived from the seasonal cycle study to the long-term trend. And you can see that both of the estimates of C13 discrimination by the terrestrial biosphere over time are about the same, about 0.25 per mil per decade for what we derived from the seasonal cycle, and about 0.22 per mil per decade from what we derive from the long-term trend. That's good that they match up. It's also not required. This method, or these two methods, are sensitive to different regions of the terrestrial biosphere. So the long-term trend method is especially sensitive to the tropics, and the seasonal cycle method is sensitive to the temperate mid-latitude region up to the Arctic. So they're sensitive to activity, ecosystem activity in different parts of the world. Of course, this C13 discrimination doesn't have much meaning for most people, so we want to convert it to a trend in CI over CA concentrations and a trend in water use efficiency. So this is just a real simplification to remind you that the C13 discrimination change is related to a potential change in CI over CA. So what we've done here, we have estimates of the C13 discrimination, and we want to solve for a change in CI over CA. I've introduced this simplified version, which is called the Linear Approximation, but there's a more precise version that's important to consider. This includes terms from the influence of mesophil conductance and photorespiration. And you can see that each one of these terms contains the atmospheric CO2 concentration in it. So if we're looking at these studies over time, for instance, from 1985 to 2017, atmospheric CO2 concentrations increased by 55 ppm. So it's important to take these terms into account whenever we're talking about long-term trends in C13 discrimination. So if we factor those in, then we solve for a change in CI over CA, and that is the information that we need to use to calculate intrinsic water use efficiency. Right? So just to point out, if we have no change in CI over CA, but atmospheric CO2 concentrations are increasing, that means that intrinsic water use efficiency will increase as well. Okay, so just to summarize then, what we've learned from the seasonal cycle and the long-term trends in terms of water use efficiency. So the seasonal cycle observations predict or estimate an increase in water use efficiency of about 14%. Over this time, CO2 rose by about 18%. And then from the long-term trend, again, this is more sensitive to the tropics, whereas the seasonal cycle method is more sensitive to the higher northern latitudes. For the long-term trend, we get an increased water use efficiency of about 11%, while CO2 rose during that period by 14%. So what this means is that ecosystems from the tropics to the temperate mid-latitudes to the Arctic all seem to be increasing their water use efficiency at about the same rate as atmospheric CO2 concentrations. So this slide over here is just a rough diagram to show you that if there was a constant Ca minus Ci value, that would mean no change in water use efficiency. If there's a constant Ci over Ca value, that means a modest change in water use efficiency. And then if there happened to be a constant Ci value, that would be definitely outside of our observations here in black and the magenta line. So all this is pointing to trends in the terrestrial biosphere of a relatively constant Ci over Ca ratio and a modest increase in water use efficiency. So the atmospheric results imply what I just said, a constant Ci over Ca values. But if you add this up over the 20th century, it means a 20% increase in intrinsic water use efficiency. These atmospheric trends are roughly consistent with trends in the tree ring records. So you can study trees by taking the carbon from the tree rings and measuring the C13 ratio of the tree rings and see how they change over time. Again, this is very analogous to what we've measured in the atmosphere. So recent publications have looked at records of northern hemisphere tree species and looked at how the C13 of these trees change over time. And they're also more or less consistent with this constant Ci over Ca trend. This is all in line with something called stomatal optimization theory, which predicts a constant Ci over Ca as atmospheric CO2 increases about proportionally. So a 1% increase in CO2 concentration in the atmosphere would result in a 1% increase in intrinsic water use efficiency. So this just assumes that plants adjust their stomatal conductance to maximize overall carbon gain, and also considering the metabolic costs of supplying water for transpiration. Okay, so as more carbon cycle models incorporate C13, these records, both the atmosphere and these example of tree ring records, provide important validation. So this is a study by Keller. On the top panel here, we have change in isotopic discrimination from tree ring records in the gray, and then the average in the black line. And they've compared it to two different models, the LPX model in red and the CLM model in blue. And you can see that these changes in C13 discrimination are related to increases in water use efficiency, and that the LPX model matches the observations a little more nicely than the CLM model. Okay, so while these overall patterns are converging on constant Ci over Ca values, different plant functional types and species differ in their water use efficiency responses. So this is a figure from Frank et al. In 2015, and here it's showing changes in intrinsic water use efficiency extracted from tree ring records for conifers in red and broadleafs in blue. And you can see that the conifer and the broadleafs are increasing their water use efficiency at different rates. And the models might not be recreating or matching those species or those plant functional type differences. So let's consider some individual tree species then. So now we're moving from the global scale back down here to the leaf scale. So looking at using C13 isotopes of plant tissues to study individual species, the gas exchange, and their intrinsic water use efficiency. Sorry, so this is work by my PhD student, Yumi Oh. We are submitting it this week. In fact, if I didn't have to prepare the talk, it would probably be in by now. But this is looking at carbon allocation in trees and how that influences intrinsic water use efficiency estimates. So there are different ways to measure plant water use efficiency. The gold standard is to use a Lycor photosynthesis system, which is basically just a leaf chamber where you clamp it on the leaf and you measure CO2 and water fluxes. So I'm going to call these the instantaneous measurements. There's also methods to estimate water use efficiency at a whole plant scale, but the other common way of doing it is looking at eddy covariance fluxes, where you measure net CO2 exchange. You can use some modeling estimates to get at what that means for photosynthesis rates, and then you can compare that to ecosystem evapotranspiration, right? And so you can get CO2 fluxes and water fluxes that way. So the community, right, has been measuring water use efficiency at these different scales and comparing them over time, and there's some offsets. So this is a meta-analysis paper by Medlin that was published in 2017, and here she's looking at publications that estimate water use efficiency for different plant functional types using different methods. So if we just zoom in here on deciduous broadleaf, we have the green are the leaf gas exchange measurements, the orange are the leaf isotope measurements, and the blue are the flux net measurements. And if you kind of peek across here, there's kind of a consistent offset between the green and the red, meaning that the leaf isotope methods tend to predict a higher water use efficiency than the leaf gas exchange. So it's not totally clear why that is. So we studied leaf C13 records from a forest here in Indiana, at the Morgan Monroe State Forest. This is a little bit south of us. This is an Ameriflux tower site, so they have eddy covariance measurements, as well as samples of four different species. So we're talking about poplar, sugar maple, sassafras, and white oak today. So the leaves were sampled from the canopy over different days through the growing season over different years. So this is a mashup of data from 2011, 2012, 2013, as well as 2017 and 2018. So what we have plotted here is day of year that the leaf was sampled, its delta C13 value, and then for the four different species. And so one of the kind of patterns that you can see is they all tend to kind of start higher on C13 and then kind of move down a little bit as the growing season progresses. These panels on the right are just some averages of the leaf C13 values and some tree ring observations as well. Okay, so this spring enrichment in C13 in the leaves cannot be explained by photosynthetic discrimination. So this dashed line here is what we would predict just from what we know about how these leaves respond, or these plant species respond to vapor pressure deficit, and how their CI over CA concentrations change with time. We would predict relatively lower values of C13 in the early season, and then maybe enrich a little bit in the middle of the growing season as conditions become drier. So what we see, this early enrichment, we think this can be explained by using stored carbon for the spring leaf flush. So it's been shown that remobilized non-structural carbohydrates from storage pools within the trees tend to have higher delta C13 values. So if the spring flush is driven by this older stored carbon, which has a higher C13, that can explain the general patterns that we see. So what Yumi has done is created a model of leaf carbon that includes a structural carbon pool and a non-structural carbon pool in order to track changes from what we're calling old non-structural carbohydrates or stored carbon to the new photosynthetic assimilates. And so her model kind of takes into account carbon coming into the leaf through photosynthesis, growth and respiration losses, phloem transport, and then remobilization of the stored carbon. And so then she predicts changes in the fraction of new carbon, both in the non-structural carbohydrate pool and the structural carbohydrate pool over time. Okay, so that's the model. Let's jump to the results. So using this stored carbon, this remobilization of stored carbon, we can explain the early season enrichment in C13 and persistent offset and intrinsic water use efficiency estimates by different methods. So what we have here, so this top panel is the mean intrinsic water use efficiency for the 4 different species, and each different color is one of the 5 different methods that we compare for measuring water use efficiency. So I mentioned the gold standard is this bright blue bar here. This is the instantaneous leaf gas exchange measurements that were done on the leaves at the field site. The cyan color is our model that's been tuned to those observations. And so we sample the model at the same time as those leaf gas observations were made. And those are typically done midday. So what's interesting then is our model has sort of high temporal variability. And so if we take an average water use efficiency that's GPP weighted, so not just sampled at the time that those leaf gas exchange measurements were made, but averaging it out GPP weighted over the whole season, we get lower intrinsic water use efficiency estimates for each of these species. By comparison, the water use efficiency estimates from the leaf C13 values are in green and the tree ring C13 values are in yellow. And we've done 2 different calculations there. I mentioned the simple linear method predicts a large overestimate of water use efficiency. And then the bar the horizontal line within that bar is the correction for the mesophyll conductance. So using that full equation, incorporating mesophyll conductance reduces this overestimate by the isotope methods, but it doesn't completely eliminate it. So we think that the allocation of remobilized carbon explains the difference between these C13 estimates or the isotope estimates and the instantaneous leaf gas exchange measurements. It's important noting that the true physiological water use efficiency values are probably lower than these midday leaf gas exchange estimates because they're biased during these midday conditions when water use efficiency is a little bit higher. And then we can look at response to drought too. So at this particular site, 2012 was a drought year. And so we can look at the difference between 2012 and 2011. And so pretty much all of these different ways of estimating water use efficiency show that water use efficiency was higher in 2012 during the drought. But the relative magnitudes and the differences by species vary by method. So if you were just looking at the leaf gas exchange measurements, right, these 2 species, sugar maple and tulip poplar, look like they increased water use efficiency the most. If you're going by the carbon isotope measurements, it would look like sassafras increased the water use efficiency the most. So we just want to note here that this process of invoking remobilized carbon affects the mean bias and probably the drought response, but not necessarily the long-term trends in intrinsic water use efficiency that we were discussing earlier. So it depends on the time scale that you're studying these things. So just to summarize this plant tissue investigation, so we looked at the role of carbon allocation in interpreting plant tissue C13 records. And the use of stored carbon can explain why these C13 methods tend to estimate higher water use efficiency than other methods, at least in the deciduous broadleaf trees that we studied. So caution must be taken when using the leaf or tree ring C13 in order to validate the magnitude of water use efficiency in carbon cycle models or looking at interannual variability. The summary from the atmospheric investigation showed that water use efficiency increased in response to increasing atmospheric CO2 mainly with maintaining a nearly constant CI over CA ratio. And this means that the global terrestrial biosphere has become less constrained by water stress over time as a consequence of increased CO2 in the atmosphere. Of course, this is a coupled system, so increased intrinsic water use efficiency influences the coupled carbon-water cycle through vegetation climate feedbacks. So there's some indication that increased intrinsic water use efficiency could relax water stress in many plants. But if you think about kind of the downwind cascading effects, if there's less transpiration, there may be less precipitation downwind, which could enhance water stress in some regions. So this vegetation climate feedback can offset some of the benefits of elevated CO2, or CO2 fertilization is also called. So this is a study that I worked with a student here publishing in GRL, a modeling study. Of course, then there are also the possibility that leaf area can increase and atmospheric demand can change over time, which is going to play a role in these trends in water use efficiency. So we've talked about a whole scale, studying carbon isotopes from the global scale all the way down to the leaf scale. Carbon isotopes are well suited to studying this process across the different scales. Questions remain, though, about how to interpret leaf and tree ring C13 and how these intrinsic water use efficiency influences on the carbon cycle and the water cycle. Okay, so moving it back then, closing on this diagram of the modeling world. So we talked about advances in knowledge of stomodal conductance, and then also advances in understanding the allocation of photosynthesis. So not only, is this important for interpreting carbon isotope records, but also studying this tree system and the C13 tracers that it provides could be a way of studying this dynamic non-structural carbohydrate pool in a little more detail as well. And so with that, I think it's about right on time. And I'm going to thank my collaborators, Ralph Keeling and Heather Graven, have been participating in the atmospheric CO2 work. And then my student, Yumi Oh, Kim Novick, and Kong Yi at Indiana University participated in the tree carbon study as well. So with that, I'll thank you and take any questions.
Thank you, Lisa.